At the start of the labs, a coprosma specimen was sampled. Throughout the rest of the labs, a series of different methods were used in an attempt to identify which species of coprosma the sample was. The first method used to try and identify the species of coprosma collected was to use morphological observations. The morphological features of the sample collected were compared against dichotomous keys. The sample was also compared to vouchers of different coprosma species which were stored in the UC herbarium. Based off the morphological observations, the coprosma was believed to be Coprosma areolata. This is due to the presence of hair on the leaf and stem, a clearly visible vein network, and a scalloped leaf margin. The next method used to attempt to identify the species of coprosma collected was to use a DNA sequence from the sample to conduct a search for similar sequences using the web page Standard Nucleotide Blast. It was found that the best match for the sequence was Coprosma huttonea. However, a preliminary search indicated that this species is endemic to Australia. Because of this, the coprosma was believed to be Coprosma rhamnoides, as it was the second closest match to the sample. The last method used to try and identify the species of coprosma was a phylogenetic analysis using different species of coprosma for the in-group and a species from the genus Nurtura as the out-group. From the phylogenetic analysis, it was found that the sequence which was the most closely related to the coprosma sampled belonged to Coprosma rotundifolia. However, the posterior probability for this clade was found to be quite low, and as such, the morphological features of the specimen were assessed to see if they supported this identification. The morphological features of the specimen which match Coprosma rotundifolia are an apiculate leaf, the presence of hairs on the branch, the presence of hairs at the leaf apex, the presence of hair on the midvein, and the presence of secondary veins. In conclusion, based off the phylogenetic analysis and the morphology of the coprosma sampled, the coprosma is believed to be Coprosma rotundifolia. The key takeaway point from these labs is that there are many different methods which can be used to produce an identification of an organism. Although the results of these methods may sometimes conflict with each other, a strong identification will combine these different methods to provide a larger body of evidence which supports the identification.